All right. So welcome again. Let's continue model 5 of BCHEM 563, BCHEM 565. So in this one we are going to look at mating type switching in yeast. Okay. So just like in human beings we have females and males. Yeast too, they have different mating types. Okay. And one thing about them is they can change their mating type or their cell type or sex type any number of times they want. Okay. Depending on the expression of certain genes. Okay. So yeast cells can be grown as either diploid or haploid, right? And obviously the mother cell is going to produce the cells that are identical to the daughter cell. So mostly they start off as diploid cells, right? So the diploid cells will either continue to grow by budding or they will be induced to undergo meiosis, which will consequently lead into haploid cells. Okay, but the formation of haploid cells is mostly the formation of spores during unfavorable conditions. Okay, so there will be, I think there will be a diagram over here. Yes. Sure. So this alpha cell or the one with the red dots and the one with the blue dots are diploid cells. Okay, so normally they will go through mitosis. Right, that's the budding stage. They will go through mitosis to form daughter cells. They can also go through mitosis, but they can also come together. That's fusion. Okay, they can come together and still go through mitosis or go through meiosis. That's the formation of haploids or spores during inconvenient conditions. Now let's look at the mating types in yeast. In mating types, we have actually two. Let me say three generally. So the two main sex types are A and alpha. Okay, but those are haploids, and we have a diploid one consisting of both A and alpha. Okay, so cell mating actually happens between type A and type alpha. The diploids are considered too big to undergo mating. Okay. The two haploid cells, the A and the alpha, cannot be distinguished like phenotypically. Okay. But they can be distinguished genetically based on their characteristics and also the type of mating partner they choose. The reason is that type A can only mate with type alpha, and type alpha can only mate with type A. Not to sound homophobic or anything, but just like in humans, males and females mate. Any male who mates with a male is, try. So yes, an alpha cell or a cell that is alpha, okay, is going to secrete sex hormones or pheromones called alpha factor that will attract, I won't call arrest, but it will attract alpha cells. But before cell mating can occur, the cell cycle has to be arrested. So it is these pheromones that arrest the opposite cell mate, okay, in the cell cycle, so that cell mating can occur. Same thing applies to the A cell as well. So if A wants to mate, it is going to secrete a pheromone which is A factor that's going to arrest an alpha cell in the cell cycle so that mating can occur. As I said, the cell cycle has to be arrested before mating can occur. So it is needed for successful mating. And as I said, also diploid cells are considered too big, okay, to mate with haploid cells, and also they do not respond to mating hormones. So these are the two main reasons why diploid cells cannot mate. And the cell type, whether the cell is going to be A or alpha, is actually controlled by a particular locus in chromosome 3 called the MAT locus or the mating type locus. Okay, so as I said, it is found on chromosome 3 in yeast, and there are actually two types, two alleles on the MAT locus. Okay, so in A cells you are going to find MAT A allele, and in alpha cells you are going to find MAT alpha alleles. Okay, for the diploid cells they are going to have both A and alpha alleles. But actually they are not expressed, so we will actually look at them, so don't worry. The mating, even though the mating type, changing of mating type is under very strict genetic control, certain strains of the yeast switch their mating types as frequently as every cell division. So every cell division they are most likely to change their quote unquote gender, right? So and in doing so they have to express the right set of genes so that everything will be coordinated. um let's look at the control of yeast cell type mating by, so let's see how a particular cell is determined, like a particular cell's gender, or let's see how the mating type is going to be determined, right? So as I said, they have a locus called the mat locus, the mating type locus, and there are like a number of structural genes present in or on the locus, okay, that are required for the production of a particular mating type, okay. And there's also another structural gene which is not found on the mat locus, but it is called MCM1. It is not on this video, you mentioned you will come across it as well. So in the mat locus also has, as I said, we have two alleles of mat locus, mat alpha and mat A. Okay, mat A is found in cell A and mat alpha is found in cell alpha. Okay, so the structural genes, which is the mat, so let's look at mat A. It is going to code for A factors or A type. Okay, it's not here, but it is responsible for the expression of A type genes. Okay, it actually expresses a protein called A1 or a factor called A1. Okay, and then mat alpha is also responsible for a cell becoming an alpha cell. Okay, that one also codes for two proteins, alpha 1 and alpha 2. These two alleles, the mat alpha and the mat A, they have different set of genes because they encode different regulatory proteins. As I said, the mat A is responsible for encoding a regulatory protein called A1. However, this A1 does not play a role in determining whether a cell is going to be A type or alpha type. Okay, so at this point we are going to look at A1 as useless, but it's not useless. It has a role it plays. So even though it is a haploid specific protein, it does not dictate or determine whether the cell is going to be A type. It is actually the MCM1 complex that determines if a cell is going to be A. Okay, so this was the MC1 complex I was talking about. This is also a structural protein or a regulatory protein, but it is not encoded by the mat locus, unlike the mat alpha and the mat A, right? So in A cells, the MCM1 are responsible for cells that become A type. Okay, so they turn on the expression of structural genes by binding to regulatory sequences within a promoter of the A specific genes. Okay, so this is it for the A type cells. So you realize A type cell will have a mat A locus, right? And the mat A locus is going to encode this protein A1. But we said A1 is not responsible for determining whether the cell is going to be A or not. It is the MCM1. Okay, that will bind to specific A specific genes and it will turn on or it will make the cell A, right? But it is different in the alpha cells. For the alpha cells, they also have mat alpha, but they produce, the mat alpha encodes two proteins, alpha 2 and alpha 1. Okay, alpha 2, because MCM1 is present in all yeast, the MCM1 has to be repressed or inactivated, because it is, if it is left on, it is going to encode A proteins. But now we want our cell to be alpha, so alpha 2 will bind to the MCM1 so that it cannot turn on alpha A specific genes. Okay, then the alpha 1 is responsible for the regulation of alpha specific genes. Okay, so in alpha cells, alpha 1 is responsible for the expression of alpha specific genes. Okay, but in A cells it is the MCM1 which is responsible. In diploid cells, all the factors have to be silent, right? So the alpha 2 obviously will bind to MCM1 to prevent A specific genes from being expressed, and also A1, which we actually concluded it was useless in the first one, will bind to alpha 2 as well to turn on. It also inactivates alpha 1 to prevent the expression of alpha specific genes, but that one is not showed here, so we'll go through it in the slides. I think I've mentioned everything over here. Um, the function of alpha 1, oh come on, the function of alpha 1 and alpha 2. Okay, in that alpha 2, that it is actually a dimer. Okay, so it binds to MCM1, and in doing so it prevents the expression of a specific genes. Okay, but the alpha 1 binds in concert with MCM1 to actually promote the expression of alpha specific genes. Okay, so this is in the diploid E cells, those that have both A and alpha. Okay, so you in the diploid ones, yeah, as I said, all the cell mating genes, the structural genes involved in cell mating are shut down. So there will be no A specific genes or alpha specific genes. Okay, so the A1 protein by which was produced by the MAT-A leucos will play a part as at last, because initially we thought it was useless. So it is going to bind to MCM1 and prevent the regulation or the transcription of A specific genes. Okay. So this is it. Okay. So actually in diploids, the A alpha 2 exists in two forms. One binding to M21 complex, which is going to prevent the activation or transcription of A specific genes, and one also binds to A1. Okay, that's going to repress the formation of haploid specific genes, right? So moreover, the alpha 1 A1 alpha 2 complex is also going to repress the expression of alpha 1 genes. Okay, so in doing this we are shutting down in doing this, we're actually shutting down all the haploid specific genes in the diploid cells. All right. Yeah. So now we've been able to determine how a particular cell is given a gender or a cell type, be it A or alpha. But before we started, we actually said that the yeast are actually capable of switching their cell types. So how do they switch their cell types? Let's do this. So there are some genes, okay, that are responsible for this. So sorry, these are the three main genes involved in mating type switching. Okay, the HO gene, HMRA, and HML alpha. Okay, so HO gene encodes for an endonuclease. Okay, and obviously this is responsible for A type cells, and this is responsible for alpha type cells. Okay, so we all know what endonucleases do, right? So the thing is, these are silent genes. Okay, so they are actually found in all cell types, whether the cell is A or alpha. Okay, so if the cell is A and it wants to switch to alpha, it is going to activate this so that it actually substitutes the A it has for the alpha here. So it is going to switch from A to alpha. But if the cell is alpha and it wants to become A, it is going to substitute its alpha with the A here so that the cell will finally become A. Okay, but in doing so, the endonuclease, which is encoded by HO, will have to be produced first, and it will go and cut the existing gene, which is the MAT locus, so be it MAT-A or MAT-alpha, before the one here or here will move and go. It won't actually move. A copy of it will be made and it will go insert itself at the MAT locus, right? Let's look at. So I think here it is. So the endonuclease initiates the mating type switching, blah 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 blah. So what it does is it's going to cut at a MAT locus. Okay, so if let's say it is a MAT-A, it is going to cut MAT-A, so the cell will no more express A specific genes. Okay, so in this case we are assuming the cell wants to turn to alpha, so it is going to activate it's going to activate HML alpha, okay, and make a copy of this, right, of the alpha specific gene, and we will send our copy to wherever the endonuclease had already cut. Okay. So this is a diagram showing the mating type switching. So this is just telling you it is not the whole HMRE or HML alpha that's moved. We just make copies of them. So at any given point, these silent genes are still present. We just make copies of them. Okay, they are never lost. And switching is reversible. Blah blah blah. These are not really important. So why are the HMRA and the HML alpha transcriptionally silent, or let's say yes, permanently silent? Why are they silent? Because they actually associate with certain proteins we call the cell proteins. Okay, cell proteins are silent information regulators. Okay, so when they bind to the cell regulators, cell proteins, they actually that's why they are silent. Okay, so in cell mutant, that's cells with their cell proteins mutated, the silencing of the HMRA and HML alpha is going to be compromised. That means the cell is going to code for both A and alpha, and that resulting cell is going to be sterile. Okay, but in the normal type, let's say the HML alpha or the HMRA, the cell proteins are going to be able to silence the two for the expression to be done on only the mat locus. So depending on the specific mat allele present. Okay, so we have sorry, we have three types of the cell proteins. Okay, we have set 2, set 3, and set 4, and they play a role in gene silencing. Set 2 is a deacetylase, histone deacetylase. Okay, as we all know, acetylation means formation of e-chromatin. Okay, so if we are deacetylating, that means we are promoting heterochromatin formation or silencing of gene. Okay, it's like as when we add acetyl groups, we are activating the gene. So if anything comes in to deacetylate or remove the acetyl groups, we are silencing the gene. Okay, so the SER2 is a deacetylase. It comes in to remove acetyl groups. So it facilitates the condensation of chromatin. Okay, or it facilitates the formation of heterochromatin. And it also helps lock up the HMRA and the HML alpha in the chromatin domains where transcription cannot be initiated. Okay, that simply means it helps silence them, right? So the silent HML and the HMR loci actually nucleate heterochromatin formation via binding of several proteins. Okay, so when these proteins come in and the propagation of heterochromatin will be initiated, similar to how telomeric regions are silenced. So let's look at the difference between gene silencing and gene repression. Gene silencing is actually an epigenetic effect. Okay, so that's like position effect, where the position of a gene is going to determine whether it's going to be heterochromatin or e-chromatin. But gene repression actually involves proteins, transcription factors. So these involve like physical effectors, the presence of physical effectors, but this is just like position effect or epigenetic effect, right? So let's look at heterochromatin formation and the maintenance and its maintenance, and how it depends on interactions with histones. So I think by now you know the cell proteins or the cell complex. Okay, it's actually analogous to a protein called HP1, just that the cell complex or the cell proteins operate in yeast and the HP1 operate in mammals. Okay, so the same function the cell proteins do or perform in yeast, that's the same function HP1 proteins or protein do, okay, in mammals, even though they are not the same proteins, but they perform the same function in mammals and yeast. Okay, so HP1 actually binds to methylated histones. Okay, and they lead to the formation of high order chromatin structures, or they lead to the formation of heterochromatin states. So methylation of histones actually creates binding sites for HP1. Okay, so if there are no methylations, HP1 cannot bind to the chromatin. Okay, so methylation of the histones is going to create binding sites for HP1 to bind. That's in mammals. and cell complex is in yeast. Okay, there's another protein called RAP1, and this RAP1 will initiate the formation of heterochromatin in yeast by binding to specific targets. So RAP1 actually works with the cell complex in yeast in the formation of heterochromatin. Okay, so for still on the RAP1, the targets where the RAP1 bind include telomeric repeat silent and silences. Okay, especially at the HML and the HMR, right? So these are the common target of RAP1, and this is how the RAP1 functions. Initially, it will recruit cell 3 and cell 4, right? Cell 3 and cell 4 are going to bind to the N terminal of histone 3 and histone 4. So afterwards, cell 2 comes in, or cell 2 is recruited, and we know cell 2 as a deacetylase. So when cell 2 comes in, it will remove acetyl groups, and this will promote the spreading of cell 3 and cell 4. Okay, there's another pathway that promotes heterochromatin formation at central base, and it is called RNA interference. Okay, so RNA interference promotes heterochromatin formation at central base. Inactivation of chromatin occurs via two ways. We know covalent modifications. We also know the addition of proteins to nucleosomal fiber. Okay, so these are some two ways where a chromatin can be inactivated. Covalent modifications like removal of acetyl groups or methylation of the histones or the DNA, blah 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 blah, right? And also the presence of some proteins. Okay. The inactivation of a chromatin can be due to the different effects. Okay, it can be the chromatin being inaccessible. Okay, so the chromatin won't be accessible for gene expression. Also, there can be proteins or the addition of proteins that will directly block the access of regulatory sites. Hence, that gene is not going to be able to express, right? And also, there can be proteins that directly inhibit transcription. So these are three ways that affect the inactivation of chromatin, or let's say that affect the formation of heterochromatin formation. Okay, so one, condensation of chromatin to make it inaccessible, okay, to the apparatus, whatever apparatus needed for gene expression. So it's like the chromatin condensing or folding up, okay, to make the particular regions needed for gene expression to occur inaccessible. Okay, and also addition of proteins or recruitment of proteins that will block regulatory sites like the promoter regions and enhancer regions, and also proteins that will directly inhibit transcription. Okay, like silences. So I've mentioned that we have HP1 and cell complex in yeast, and they are involved in heterochromatin formation. So let's look at HP1. Okay, HP1 is heterochromatin protein 1, and it is a type of SUVA proteins in Drosophila. Okay, the main gene is SUVA25, but it is now called HP1 alpha because they recently discovered other proteins called HP1 beta and gamma. Okay, so the normal HP1 that's involved in heterochromatin formation is actually called HP1 alpha now. Okay, and this is the point of contact or the point of interaction with a chromosome is called the chrome shadow domain. So on a chromosome, HP1 binds to a place, and that place is called a chrome shadow domain. That's the green part of this structure. So the HP1 chromodomain will bind to histone 3. Okay, and that histone has to be methylated, either dimethylated or trimethylated at lysine 9 residue. Okay, so as I said, methylation provide or produce binding sites for this HP1. So if there are no methylations, HP1 cannot bind. Okay, so specifically they bind to di or trimethylated lysine 9 residues. Okay, so if that particular cell do not have deacetylases, okay, that means acetyl groups are not going to be removed, hence methylation is not going to occur. Okay, so if all these sequence of events happen, That means HP1 cannot bind. Okay, because we need a deacetylase to remove acetyl groups on K14, which is lysine 14. When this deacetylation occur, methylation at K9 or lysine 9 will occur. Okay, and when this takes place, it will result in the formation of binding site for the HP1 to bind. Okay, so when there's a mutation, when there's a mutation in the deacetylases, HP1 cannot bind to chromosomes. So this is just an explanation. So deacetylases are going to remove acetylations at K14 and will allow a type of methyl transferase called the SUVN39H1 to methylate K9. Okay, so this methylation is what is going to create the binding site, okay, for the HP1 to bind. So when the HP1 binds, it is going to cause an inactive region. Okay, so the inactive region can extend, okay, by the ability of the HP1 molecules to interact with one another. Okay, so it's like propagation of heterochromatin states. So therefore, the binding of the HP1 to methylated histones will form or act as a trigger for silencing, because additional molecules of HP1 are going to aggregate and interact with each other. All right. So yes, methylation of lysine 9. Take note of this. Methylation of lysine 9 actually is responsible for heterochromatin formation, but methylation of lysine 4 is responsible for echromatin formation. Okay, so even though they are all methylation modifications, this is on lysine 9 and this is on lysine 4. Methylation on lysine 9 results in silencing or heterochromatin formation, but methylation on lysine 4, that's the opposite, which is the formation of echromatin, right? The link between K4 demethylation and K9 methylation suggests that the two reactions act in a coordinated manner to control the relative state. So when we are demethylating K4, we are actually forming heterochromatin. I'm sure you understand this, right? And when we methylate 9, you're also forming heterochromatin. And that the link between the demethylation of K4 and methylation of K9 suggests that these two reactions act in a coordinated manner to control the relative state of heterochromatin or echromatin of a specific region. So let's look at the cell genes or the cell complex, right? The cell proteins, just like the HP1 we've looked at. Heterochromatin formation at telomeres and silent mating type loci in yeast relies on overlapping. So the cell genes or the cell complexes play a role in mating type switching in yeast. The silent mating type loci, the HMR and HML, right? The silencing of these genes, and also the formation of heterochromatin at telomeres, okay, in yeast, to be specific, in yeast. The binding of the cell proteins can actually silence any promoter or coding region. So we actually know the cell proteins to be silencers, right? So if there is any mutation in the cell proteins, obviously their functions are going to be their functions are they are not going to perform their functions, right? So it's going to be like the opposite of whatever they do is going to happen. So normally in the silencing of the mating type loci in yeast, for example, if there are any mutations in the cell genes, the HML and the HMR are going to be activated instead of being silenced, right? So therefore, the product of the cell genes or whatever proteins the cell genes produce maintain this inactive state of the heterochromatin or the HMR and the HML. This is not really important. So I think I've mentioned earlier that in yeast, cell proteins or the cell genes work in conjunction with RAP1. Okay, so RAP1 actually nucleates or initiates everything. So the first thing is RAP1 will bind to a C13A repeat at telomeres. Okay, so this is silencing at or heterochromatin formation at telomeres. So the RAP1 will bind to this repeat C13A repeat. So and it also binds to cis actin silencer elements that are needed for the repression of the HML and the HMR. Right. So the RAP1 is involved in heterochromatin formation at telomeres and also silencing of mating type loci. Okay. In the telomere, the cell3 and cell4 will interact with RAP1 first before the cell2 proteins are recruited. Okay. So the cell3 and cell4 come in and they interact with the N terminal of histones 3 and 4. Okay. Specifically for unacetylated tails, because we know unacetylated tails means it's unacetylated tails means it favor heterochromatin formation. Okay, so when these two are in position with with the RAP1, the cell2 are recruited. Okay, to further remove acetyl groups that are nearby, and this is going to promote the binding of cell3 and cell4. Okay, and be spreading the heterochromatin formation. A similar series of events forms the silence. So what we just described is formation of heterochromatin at telomeric regions. Okay, RAP1 will bind to cell3 and 4, and they interact with the N terminal of histones. Its preference for unacetylated regions before the cell2 comes in to further deacetylate neighboring acetylated histones. Okay, so similar thing occurs when we are silencing the HMR and the HML genes. Okay, so and that one, even though they are similar, it's not like exactly the same factors. So in telomeres, from heterochromatin formation at telomeres, it was only RAP1 that will recruit the cell2, cell3 and cell4. But in this one, there are three in silencing these two genes. There are three sequence specific factors that are involved in triggering the formation, not only RAP1 but EBF1 and ORC. Okay, EBF1 is a transcription factor and the ORC is called origin replication complex, and I'm sure we come across this before in the early models. So, and this one also includes cell1. Okay, formation of heterochromatin at the telomeric regions didn't include cell1. It was only cell2, 3 and 4. Okay, so the first thing that happens is cell1 will bind to the sequence specific factor. Okay, I don't know which one of them, but when it binds, it's going to recruit cell2, 3 and 4. Okay, to form a repressive structure. And we know the function of cell2, the deacetylation, and it is very necessary to maintain a binding of the cell complex to the chromatin, because we know the cell complex or the cell genes bind to histones with preference for unacetylated tails. Okay, so to keep the cell protein still bound, we need the cell2 to be removing acetyl groups to keep the cell protein still bound. Okay, so this is the summary. The RAP1, the RAP1, this is I think in telomeres, right? RAP1 will bind or identify the sequence, okay, wherever we are going to form the heterochromatin. So let's say at the telomeric region, RAP1 will identify the region where we are going to form the heterochromatin. Then we'll recruit cell 4, cell 3, then cell 2, which is an HDAC. HDAC is histone deacetylase. Okay. We've gone through all of this. So these are two ways a gene region or a region can be inactivated. That's coating with the cell3-4 complex itself has an inhibitory effect. Okay, and also the function of cell2 also has a further inactivating effect on the region. Okay, so the first step, which is the recruitment of cell 3 and cell 4 to bind to RAP1, on its own is an inhibitory effect. Okay, so it actually inactivates the region to some extent before the cell 2 comes in to do the deacetylation, and the deacetylation on its own is another inactivation effect. Okay, so the C terminus of the cell 3 proteins have similarity to nuclear laminar proteins, that being so let's look at the last one, which is RNAi dependent heterochromatin formation. Okay. The RNAi is RNA interference. Okay, so in RNA interference, it's just like using double-stranded RNA. So you see RNA actually single-stranded. Okay, so we just, it's like an RNA that has how do I describe this? If a single strand RNA, if there are complementary base pairs on that single stranded RNA, the RNA is going to form secondary structures, right? So it is these secondary structures, or this double stranded RNA molecules, that we use in degrading or inactivating mRNA. So let's say degrading. So when you are using these double stranded RNAs to degrade other mRNAs, we call it RNA interference pathway. Okay, so so let's look at, I don't know if that's what they use there, but that's what RNAi pathway means. Okay, so let's look at RNAi dependent heterochromatin formation. So the formation of heterochromatin, that depends on RNAi. The formation of heterochromatin in, I don't know if it's Saccharomyces, but S-POMB utilizes the RNAi dependent pathway. So I think the three main ways, you have to know when and where they are required. Okay, so not even the when, but where. So we have three pathways. We have three whens. Okay, we have two whens and one where. The first when we have three whens. So let's use whens. We have three whens, right? Formation of heterochromatin at the telomeres, formation of heterochromatin in S-POMB yeast. Okay, and also silencing of HML and HMR, right? Each one of them involves like silencing a particular region of gene. Okay, but they all have different like steps. Okay, so the formation of heterochromatin at telomeric region was RAP1 binding to cell 3, cell 4, then recruit cell 2, right? But silencing of HML and HMR involved three sequence specific and sequence specific. Yes, sequence specific factors. The RAP1, ABF, and the ORC. Okay, and also cell 1. Okay, so the next one is heterochromatin formation in POMB. Okay, which is the RNAi. So this pathway is initiated by the production of the siRNAs. I've forgotten. Oh, Charlie, my mind is clouded. I've forgotten the full meaning of this. I know it, but I've forgotten. So we use the siRNA. It's something. So the product, this RNAi pathway, RNAi pathway is initiated by the production of the siRNA molecules, okay, resulting from the transcription of centromeric repeats. So the siRNAs are going to form this RET complex, the RNA-induced transcriptional silencing. Okay, the siRNA component are responsible for localizing the protein. So I think this is just I think like model 3 or model 4, where we are using siRNAs to degrade a particular. So this is not actually, this is like the second one, to repress or silence a gene. Okay, so you have a long RNA, then you use dicers to break them into fragments to form the siRNAs, right? Then we assemble a RET complex, including the RET itself, ago proteins, which is the agonot, and the siRNA to form a complex, right? So this complex will bind to the region where we want to silence or we want to form a heterochromatin, and the siRNA component of the complex is responsible for localizing the protein or detecting the specific part where the complex is going to bind. Okay, so when it binds, then they are going to recruit DNA methyltransferases and histone modifiers or histone methyltransferases. Okay, and these are going to repress or promote the formation of heterochromatin. Okay, So the complex contains proteins that are homologs of those involved in heterochromatin. Examples are gonus protein. Okay, we've talked about this before. It is also involved in risk remodeling complexes to chromatin as well. Um, there's siRNA complex promotes methylation of K9 by this factor called CLR4. Okay, the CLR4 is a methyl transferase. Okay, a type of KMT1. So when the siRNA promotes methylation of K9, obviously we are moving towards heterochromatin formation. Okay, but in that diagram in model 3, you realize after the siRNA binds to RNA or the region of interest, the DNMTs are going to be recruited, and this is an example of the DNMTs or the HMTs, histone methyl transferases, that are recruited to methylate the region. Okay, so the methylation done by the CLR4 methyl transferase is going to recruit a homolog of HP1 called SWI6. We've come across this, but I didn't mention it. Sorry. So you see, we have HP1 that forms heterochromatin in mammals. Okay, and its analog in yeast in general is the cell proteins. Okay, that one is also responsible for the formation of heterochromatin in yeast. But there's a specific type of yeast called the pombe. Is the sacro just say Schizosaccharomyces pombe. Okay, so another homolog of it is called SWI6, and that one is found in only Schizosaccharomyces pombe. Okay, and they also perform the same function as HP1 or the cell complexes. That's formation of heterochromatin. So SWI6 is responsible for overseeing all these things. So why does a silencing complex repress chromatin activity? How does a silencing complex like the siRNA plus the RET and the argonaute and everything, how does it repress a chromatin activity? So one way is it could condense the chromatin. Okay, I think we've mentioned this as well. It could condense the chromatin so that the regulator proteins cannot find their regulatory point. Okay, also the presence of the complex itself is mutually incompatible with the presence of transcription factors. So when the silencing complex is available, transcription factors cannot bind to the protein and cannot bind to the chromatin. Okay, so it's like competitive inhibition. Okay, hence transcription cannot occur. Okay, and this also leads to silencing. Also, the silencing complex are going to block chromatin remodeling. Okay, so like you see, chromatin remodeling is when the chromatin is turning from maybe a heterochromatin to echromatin, if I'm not mistaken, right? In the presence of the silencing complexes, the chromatin cannot convert from heterochromatin to echromatin. Okay, unless the silencing complexes are removed. And another way is the silencing complexes directly obscuring the binding sites on DNA. Okay, so it's either the chromatin is going to be condensed to prevent regulatory proteins from functioning, or competitive inhibition between the silencing complex and transcription factors, and also a RNA polymerase, or the silencing complexes blocking chromatin remodeling and also obscuring or blocking binding sites for transcription factors. But even though we said there is a competitive inhibition, there's a competitive inhibition between the silencing complex and the transcription factors, sometimes we can still find transcription factors at silenced regions. My friend is saying, aside the presence of transcription factors and the polymerases are promoters of silenced genes or silenced chromatins. Okay, and this implies that the silencing complex prevent the factors from working rather than them coming in just to bind to their promoter. So it's not like, so this what is it, this point does not mean that if the silencing complex is available, the TFs cannot come in. Well, you can come in, but you cannot function, right? So that's how the silencing complex repress chromatin activity in that sense. Competition between the. So let's look at heterochromatin formation at the centromere. So we've looked at heterochromatin formation at the telomere, which is the RAP1 binding to cell 3, cell 4, the recruit cell 2, right? So what of heterochromatin formation at the centromere? So in human cells, we have a centromeric specific protein called CNPB. Okay, and that protein is required to initiate modifications of histone 3. Okay, these modifications include removal of acetal groups of K9 and K14. Okay, and also methylation of K9. We know the implication of this: removal of acetal groups and addition of methyl groups. Okay, all these are just done to promote heterochromatin formation. So take note, at the centromeres we need a centromere specific protein, a protein called CENP-B. Okay, so that protein is required to initiate modifications, okay, at on histone 3, and the modifications are deacetylation and methylations. So when these modifications occur, they trigger association of the CENP-B protein with HP1. Okay, so it's like these modifications are going to indirectly create binding sites for HP1. So when these modifications occur, HP1 then interacts or binds to the CENP-B, and this will lead to the formation of heterochromatin in the region. So HP1 in mammals, right? HP1 was involved in formation of heterochromatin at telomeric regions. It's also involved in formation of heterochromatin at centromere, but the other factors are different. Okay, the telomeres included, it was just a HP1, okay, but it needed methylations before it bind, right? But at the centromere, it requires a protein, the CENP-B protein, before the HP1 can bind. So moreover, the heterochromatin and RNAi are required to establish CENP-A at centromeres. So there's another protein called CENP-A. So this one is not actually related to CENP-B. CENP-A is a histone variant of H3. So this is like a histone protein, and this one is very important to the centromere. If there is no CENP-A at a centromeric region, there will be no centromere at all. Okay, so a centromere can afford to lose CENP-B, but it cannot afford to lose CENP-A. So a CENP-A is just like H3, like histone 3. It's like a variant of it. Okay, so heterochromatin is often present near CENP-A chromatin. Okay, and the RNAi directed heterochromatin flanking central kinetical domain is required for kinetical. So these are just regions required for the assembly of kinetical. Kinetical is also found at the centromere. That's where the spindle fibers bind to during mitosis, blah blah blah. Okay, so there are other factors like this methyl transferase, the SUV39 methyl transferase, HP1, and the component of the RNAi pathway that are required to form a CENP-A chromatin. Okay, so this is just like forming heterochromatin near a CENP-A chromatin. Okay, so we need all these factors: methyl transferases, HP1, and the component of the RNAi pathway. Okay, let me end here. The next one we take it from the polycombin and trithorax